Okay. So, good evening, everyone. So today was the NPTEL TA session 11, and so today was in week 12 was uh so week 12 was a summary session. So before we got, so I thought I would do the summary session in the last TA session. That would be next week, just before the exams, which is I think on 30th April. That would be more helpful. So I thought for this part in this course. So I discuss a little since we are discussing about homeostasis. So I thought we should discuss about cellular signaling or hormone signaling, whatever you say. So before we start the before we go to the summary session, so this is kind of an extra thing which I would, because I thought it would be incomplete if we don't go to cell signaling into a little uh, deep if we are talking about basics of biology. Okay. So yeah, so we'll start this. So today's I'll do some the board work, so I'll not present the slides. So what we are talking about was the homeostasis, correct? So how was homeostasis was working? Fine. So how was homeostasis? I was just going to do. Okay, this is the eraser. Okay. Okay, fine. Okay. Uh, Primordial. Good. Uh, so, so you're thinking about homeostasis, right? So we're talking about homeostasis. So homeostasis, there was extrinsic regulation by nervous system control and the endocrine control. Now, it was not very clear, very um, detailed. There's not a very detailed discussion that how actually the hormone works inside the cells, because many of the hormones doesn't go inside the cells as it was mentioned in one of the slides, and many of the hormone um not only not and many of the hormones do go inside the cells, but there are so many hormones in the body, and there's so many metabolic reactions are going on, and hormones are controlling so many metabolic reactions. So how they are being regulated by this different kind of hormones. So interestingly, there are, so in basic hormone in basic any basic hormone action, we will always have uh, a kind of a basic generic pathway. So if I just don't know how to make title of this slide without this. So let's write basic hormone signaling scheme. Yeah. So we'll have here two types of hormone signaling schemes, correct? So so if I divide the hormone signalings in two parts, so there will be two types of hormones. One will be the hormones that the extracellular hormone, the extracellular hormones, and one will be the intracellular hormones. The extracellular hormones, most of this protein hormones or protein hormone, like protein hormones, they belongs to this kind of extracellular hormones, correct? Okay. So protein hormones for the glycoprotein hormone. So they belong to this type of hormone category. Okay. On the other hand, steroid hormone belongs to this category, which are they have the hormones that have extracellular receptors, hormones that have intracellular receptors. Uh, rather I should say not intracellular hormones. Rather, I should say intracellular receptor hormones. So hormones that have intracellular receptors or hormone that have extracellular receptors. Okay. So there are two types of hormones. Now, what is the basic difference of between this hormones? Now, if I just consider uh, this as my cell, okay, this is my cell wall, and here inside I have a nucleus, okay, and so maybe this is my cell, right? Is the cell membrane, cell membrane, and this is my cytoplasm, and this is my nucleus. Something. This is my nucleus. Now, if we think like that, this is my nucleus, this is my hormone, and I can draw a same thing here also. Okay. How does two differs? Okay. So for extracellular hormones, so the hormones will be so this is they say this is it doesn't even know how to do the undo here. Okay. Okay. For extracellular hormones, a shortcut actually. Firstly, it was there. Better. So to go back here, draw. Yeah. Um, so yeah. So now what we should do is what for the extracellular receptors, the if these are my hormones, okay, uh, so if these are my hormones if this is my hormones here, and also for here and here, if these are my hormones, the hormones receptors, so they will have some receptors here. Okay, we'll have some receptor. Yes. In the other case, in this case, the receptor will present here in the cytoplasm or in the nucleus. So what will happen? The hormone will go, the hormones will go and bind to this receptor here. In this case, the hormone will go inside the cells and bind to this receptor. So what will happen in this case? In this case, the receptor binding will be here. In this case, the receptor binding will be here. For these cases, generally, the hormones, sometimes the hormones which have hormone response elements, have I not put it? Yeah, I have put it here. So in this case, sorry. In this case, generally, these are the H hormone receptors. So in this case, in generally, the receptors in the nucleus, there will be DNA, right? Here also, here also. Correct. So there will be DNA. In this case, the hormone receptor generally itself goes and binds there to induce gene expression. So transcription means. In this case, there will be something known as second messengers. Second messengers. These second messengers will go with some of the intermediate pathways, which will ultimately bind and start the transcription. Now you can see there is a subtle difference in these two pathways. So first of all, first one has some second messenger pathways and doesn't go inside the cells. But here, the hormones go itself inside the cells, and the hormone receptors can go directly to uh, go bind the DNA and start gene expression. So what is the need of this second messenger system? Now, if you think of second messenger systems, that, for example, if you think of something like this, so if one molecule, if one molecule of hormone can bind to one set of receptors or one receptor, and if that receptor goes and binds to do transcription, there is 1 to 1. There is always 1 to 1 ratio. So one molecule of hormone can can uh, can activate only one receptor, which will do the transcription from the DNA. Now think of the other one. If one molecule of hormone bind to one receptor, if this one receptors can activate 4 second messengers, the hypothetical scenario I'm saying, then these 4 second messengers can again, so 4 of the second messengers can, if each of them can again activate 4 transcription factors or 4 some intermediate molecules. So 4 into 4, total 16. Say I just say here only these don't have any intermediate. So 16 transcription factors are there. So then the rate of transcription will be much more higher. Here, if the transcription rate is this, maybe here the transcription rate is 4 times, or the activation rate is 4 times. Not only that, this 16 transcription factors can activate then. These can only activate maybe one transcription factor, one type of transcription factor. Type of transcription factors. However, these, on the other hand, may activate maybe 4 types of transcription factors together. So as you can see here, there is a uh, clear scheme of something is very commonly known in the hormonal field, is known as signal amplification. So with one molecule of hormone now can then activate multiple transcription factors. That's why it's amplify the signal. That is something known as signal amplification, which is very important for the like this, right? Amplification by second messengers. Okay. This is a particular scheme of the hormone action, which is again described in this slide. As you can see here, if I present this, and as you can see, there is a one molecule of ligand. So each molecule of ligand activates many molecules of G protein. Correct. So G protein is kind of a receptor, which I'll come later. Which again, this G protein can amplify multiple adenylyl cycles. So then each CMP molecule activate adenylyl cycles. So now each CMP molecule can activate one molecule of protein kinase. So then four CMP molecule can then activate like multiple protein kinases. Multiple protein kinase now can activate multiple phosphorylate multiple enzyme, and thus amplification is much higher. So from one molecule, we can amplify the signal to a higher level. So that is also known as the second messengers activation by second messengers. So now we'll go to the basic scheme. So again, the basic scheme of a hormone signal will be always. So there will be a hormone in general. So we always have a hormone. That hormone will bind to a receptor. A receptor will bind to some of that second messengers. The second messengers will activate one, two, many intermediate 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 let's provide the remainder. Okay. One to many intermediates. Intermediates, which will then activate the why it is coming like okay, what is this one? This will not come. Still it is coming. So it will activate so one to many intermediates, which will activate the gene transcriptions. Okay. This is the basic scheme of the hormone receptor. So first hormone will go is the known as the G protein coupled receptors, or known as GPCRs. Okay. So how does GPCRs work? Okay. Now, as you can think of G protein coupled receptors, let's make some new slides so that it will be easy for these transitions. Okay. So as G protein. So G protein is here is GTP. So G proteins are proteins. So G proteins are proteins. What are G proteins? What G proteins? So G proteins are proteins that are bound to guanosine triphosphate or GTP. So these are known as the G proteins. What are GPCRs? Is the G protein coupled receptors. So GPCRs are G protein coupled receptors. Now they are also known as 7 transmembrane receptors. Why it is called 7 transmembrane receptors? So this is because these GPCR molecules, if this is my cell membrane, these GPCR molecules, if this is say my hormonal receptor, they have 7 transmembrane domains. 5, 6, 7. So they kind of have 7 transmembrane domains. So these are the transmembrane domains. So that's why they are known as the transmembrane domains. So that's why they are also known as the 7 transmembrane receptors. Now, generally these G proteins, so G proteins are made of 3 subunits. 3 subunits. There'll be G alpha, G beta, and G gamma. In general case, in general, in most of the cases, the in most of the cases, this G alpha proteins, very yes, this G alpha, well, let's present it and do so it will be easy for me. In most of the cases, these G alpha proteins, they are bound to the GTPs. Okay. In most of the cases, G alpha protein is bound to the GTPs, and G beta, G beta, and G gamma are generally form as a dimer. They generally act as a dimer. So this G alpha subunit, has a intrinsic GTPase activity. What do I mean by intrinsic GTPase activity? So what I mean by that is that if a GTP is bound to a G alpha subunit, or like if it is like bind like this, if it is a GTP which is kind of bound, so this is a GTP. It has an intrinsic GTPase activity to form a from GTP to maybe GDP. Okay. So what is the this is kind of the basics of GPCRs. Now, how does GPCRs work? So it works as follows. If this is my cell membrane, so I'll just draw the cell membrane again. I draw the cell membrane. So in general, so remember this is again, this GPCRs are very diverse. So most of the hormones and most of the signaling pathways are GPCRs. And indeed, there are around 70% of the genome that or the genes that actually codes for different GPCRs. So you can think of the diversity of the GPCRs it has. Okay. So what is the general theme how the GPCRs work? So if this is my receptor, this is my GPCR. Okay. So this is in the hormone. This is my GPCR. And this is my ligand. Say in this case, sorry. So in this case, maybe uh, the ligand is thyroid stimulating hormone or TSH. It's one of the exemplary hormone. So this TSH, when it binds to the GPCRs, there are several paths now path GPCRs. Now that these GPCRs are bound to the G proteins. So G proteins here I have, as I told, G proteins has alpha, beta, and gamma. Okay. And generally this uh, G protein will be uh, bound to a GTP. Okay. Now, as soon as the ligand binds to the GTP, now there will be some effector molecule. So here will be one of the effector molecule. Maybe it could be transmembrane, it could not be transmembrane, but let's for the time being, let's think of this is my effector molecule. Okay. If this is my effector molecule, from here, the two things happen. So one, the G alpha GTP complex goes and activates this complex. Okay. Now, since I have told you that the G alpha subunits have an intrinsic GTPase activity, so what it will do is intrinsic G alpha, what it will do is it will activate the effector molecules and become inactive by hydrolyzing the GTP to the GDP. Okay, so that's how it is inactivating the. So this was active G alpha subunit. This is inactive G alpha subunit. Okay, and I have told you there are one more dimer. So G beta G gamma dimer look like this. G beta G gamma dimer. They will also have some effect. They will also activate some effector molecule. And which also activate molecule. And then after that, there are several proteins that will help in reforming the subunit. So there are something known as the mm, mm, There's some problem. I'll just keep. Yeah, I think there was some problem. So yeah. So there will be molecules known as RGS molecules or receptors of G protein signaling. They will enhance this kind of this inactivation of this GTP. Now again, these things will go and these things will go. Then from the sorry. So yeah. So this will come and again this inactivated G alpha G beta subunit comes. So one of the inactivating molecules of the G alpha G beta subunit is also known as beta arrestin. So then we have a inactivated GTPase complex which have a G alpha GDP and then G beta and G gamma. So this G beta G gamma complex again gets activated by GF or guanosine exchange factors, which helps in again uh, reforming the subunits. Now remember, also there are GRKs, G protein receptor kinases. They what they do? They desensitize the receptor by phosphorylation and receptor internalization. Okay, so this is how this is the kind of the basic scheme of how GPCRs work. And so I'll give a specific example of a second messenger. So what we had here was a GPCR here. We had one GPCR. We had one thyroid stimulating hormone, which is binding. And we had one G alpha, G beta, and G gamma. What happens when this gets activated, which has a GTP, correct? So when the ligand binds, we have a effector molecule. In our case, the effector molecule is the adenylyl cyclase. What does adenylyl cyclase do? So adenylyl cyclase, this enzyme forms AMP or adenosine monophosphate to cyclic AMP, cyclic AMP, or also known as PM. Now what happens? So the activated G alpha GTP okay, by the way, I should write one thing here. I should write here. This is the uh, G. I should write insert symbols. G alpha How do I go subcranium? This is G alpha. So I'll clear later. G alpha S signaling. Okay. So this is G A, not A. This is alpha. Okay. This is G alpha S signaling by the way. So this cyclic AMP. Now there is another intermediate molecule is known as known as protein kinase A. Now this protein kinase A is a tetrameric protein. Protein kinase A. It's a tetrameric protein which have two catalytic subunits and two regulatory subunits. Using this catalytic and regulatory subunit, so the cyclic AMP goes and activates this protein binds to this protein kinase A by dissociating the regulatory complex, the R complex, from the cyclic AMP. Should write capital AMP anyway. Cyclic AMP and the catalytic subunits of the protein kinase A, the activated protein kinase A, inactive is active. Active protein kinase A goes and binds to some protein known as CRE. So with what protein kinase is a kinase. So kinase means it's phosphorylates, correct? So this protein kinase A is binds to something known as CREB. Cyclic is yeah. So CREB is a transcription factor. So the CREB, so this is the CREB maybe, which I'll just write with the different color. So this is the CREB, the inactive CREB. This is the inactive CREB. So what is this? This is CREB. So this inactive CREB is activated by phosphorylation by protein kinase A. So now this inactive CREB gets activated by phosphorylation. This goes and goes inside the, so if this is my nucleus, the nucleus, I'll have DNA molecules like this. Okay. So this CREB is phosphorylated. It's go inside the nucleus and activates the gene expression. Okay. So this is how the G alpha S or G alpha stimulation signaling goes on. Now remember, there is another signaling is going on here, which is independent of the G alpha. So there is a G beta gamma subunit, right? G beta, G gamma. So what does G beta and G gamma, G beta, G gamma do? So yeah. So what does G beta, G gamma do? So they go on and activates another effector molecule known as PLC gamma or phospholipase C gamma. Okay, so they go on activating phosphorylase C gamma, which uh, which is a phospholipase C. So it cleaves the phosphoproteins. Sorry, phosphatidic acid. The phospholipids. Yeah, it cleaves the phospholipids. So the phospholipids cleaves is known as phosphatidyl inositol. So it cleaves phosphorylate inositol to IP3 and plus DH. So this IP3 goes and activates calcium signaling, activates calcium receptors to form release of calciums influx, and DAG with some other intermediates like some transcription factors will go and bind again genes transcription of some other genes to promote growth. So these are the schematics of G alpha S signalings, and there are also two more signaling pathways or known as G alpha I and G alpha Q. Yeah. And if I just if I go to a little earlier here, okay, so I'll just so yeah. If I go here and present these things. So see, this is how the GPCRs work, and this is the G alpha S signaling. So in the next slide, I have shown you how that analytic cycles work. So as you can see, this is the adenosine triphosphate. Now this adenosine triphosphate, in absence of adenyl cyclase, the ATP, there are two pyrophosphates goes out, and the cyclic AMP is formed. So this is the cyclic AMP, whereas you can see that so as you can see here, there is a bond between the 3' OH with the phosphate bond here. So that's why this is a cyclic AMP. So this is the cyclic bond. So I'll just do a so this is how this GPCRs work, the alpha S signaling. And G alpha I signaling is nothing but the opposite of this stimulus, where instead of activating the um, G alpha subunits, it's sorry. Okay, one thing. So this was one of the G alpha S as well as G alpha Q. So in case of if it is a G alpha instead of G alpha S, sorry, my bad. Um, so for this, so if it is a G alpha Q signaling, so which is going like this. If it is the G alpha Q signaling, which will go like this. So if this is a G alpha Q instead of G alpha S, so G alpha this is very the G if it is a G alpha Q signaling instead of G alpha S, so G alpha Q GTP goes and activates the uh, PLC gamma, and which finally activates like IP3 calcium receptors and DHE. Okay, and so this is how the whole thing is shown. As you can see here, that the G protein coupled receptors, there will be alpha beta gamma subunits where the GTP is like GTP exchange is happening. And as you can see there, the alpha GTP is activating the effector molecule, in this case the adenylyl cyclase. The adenylyl cyclase then activating the cyclic AMP. The cyclic AMP is activating the inactive protein kinase A to become the activate protein kinase A. And this is cyclic AMP and the activated protein kinase A then uh, phosphorylates transcription factor CREB, which recruits its associated protein CBP or cyclic AMP receptor CREB binding protein. Sorry, and so CREB full form is cyclic AMP receptor binding element. Okay. So then the CREB with CBP goes and binds to the DNA, recruits the transcription factor TF2B, and activates the gene transcription. And as we can see the G alpha Q signaling, the G alpha Q, it activates the phospholipids P gamma or PLC gamma. Now I'll come to also tyrosine kinase receptor. So as you in the next slides, so as you can see the receptor tyrosine kinase also activates PLC gamma. This kind of give you a flavor of how signaling crosstalks or how the different hormones crosstalk with each other in the cellular milieu. So then the phosphatidyl inositol, the fatty acids, is cleaved to IP3 and DHG, diacylglycerol, and this IP3 and this diacylglycerol binds to protein kinase C to activate gene transcription. The IP3 goes and binds to goes and binds to the calcium receptor or the the IP3 receptor in the endoplasmic reticulum, which cause influx of calcium at the cytoplasm, and this calcium can again acts as a second messenger to um, induce different kind of signal. So this could be part of calcium calmette signaling and many other signaling, which I'll not go into details. Okay. So next I'll come to the second receptor, which is known as the receptor tyrosine kinase. So yeah. So till now about GPCR, do you have any question on GPCRs? Okay. If there are no questions on GPCR, so I'll move forward to my second topic, which is receptor tyrosine kinase. Or popularly known as RTKs, short. Okay. Uh, okay. So how does receptor tyrosine kinase work? So as you can think of with the name, the receptor which have a tyrosine kinase activity. Do I mean by tyrosine kinase activity? So, so if again I draw the membrane, okay, and in the membrane we have a tyrosine kinase molecule, like sorry, the receptor tyrosine kinase. So these are my RTKs. Okay, RTK. So there are two RTKs. Why two RTKs? Now two RTKs. So one of the molecule that so one of many growth factors actually have this kind of molecule. So one of the growth factors is, for example, uh, VEGF or vesicular endothelial growth factor. Vesicular endothelial growth factor, this helps in the angiogenesis of the in the cell. Okay, angiogenesis means the formation of the new blood vessels. So whenever there is a organ forming or some organ is getting damaged, new blood vessels should form, right? So VEGF helps in, or you are, or think of your child is going from a baby to a adult. So the body volume is increasing. So the uh, blood, the veins and the arteries should also increase, right? So VEGF-A, this helps in forming new blood vessels. Okay. So this VEGF, say this binds to the RTK. Now what happens if two molecules of VEGF binds to the RTK? The receptor is dimerized. Okay? So receptor is dimerized. This is my say the ligand VEGF, and the receptor dimerization is happening. So this is my just writing as V. Okay, and this is my receptor. Now these receptors have some domains. Have some domains like this, which are these domains are known as SH2 domains. Okay. So this SH2 domains, uh, this name was so the full form is SRC homology domain. So this SH2 domain there are several proteins which also have, now there is a protein, say this is my protein, which also have a SH2 domain. Now, some tyrosine molecules in this, uh, in some tyrosine molecule in this SH2 domain is phosphorylated. So the receptors are transphosphorylated or cross-phosphorylated. Means this receptor phosphorylates this one, this receptor phosphorylates this one. It's a transphosphorylation of in the SH2 domain. Now this protein here goes and binds to this phosphorylated tyrosine motif through the interaction of this SH2 domain. Now these proteins, they are generally multi-domain proteins. So this SH2 proteins also will have SH3 domains. This is also something SH3 domains. Now, this SH3 domains, there may be some other proteins also can bind here. Some other proteins can also bind here, which also have this SH2 domains. Okay. Now, this SH2 domain containing proteins is bind here. One of the such example of a protein is GRB2. What does GRB2 does is GRB2 recruits another protein, okay, here known as SOS. Okay. This SOS have a, so this is a GTP exchange factor. Okay. So the inactivated RAS GDP, which is again a small protein which activates some pathway, is now activated to RAS GTP. Now it cannot be a constitutive signaling. So a gap or a phosphatase, known as also gap, so the full form, yeah, GTP is activating proteins or gaps. They again deactivate the RAS. So as you can see, this is a constant feedback loop is going on. So once a looping is going on, so RAS is activating. So this RAS GTP activate the MAP kinase pathway, which is RAF, or known as MAP kinase, which will go and bind to activate MEC, which is basically MAP kinase. The MAP kinase will be active at ERK, which is a MAP kinase, and this will activate transcription, some effector proteins like Janus June, June FOS, AP1, like that. Okay. So this is the so this pathway is also known as this part of the pathway is known as the MAP kinase pathway. Should write the full form. Oops. I by mistake deleted all the whatever I had written. Anyway, doesn't matter. So yeah, I have it here. So this pathway here is also known as the MAP kinase pathway. It's the MAP kinase pathway. So this case, this activates the MAP kinase pathway, and PI3K also it activates another what is known as PI3K, which activates another uh, pathway, which is PIP to PF3 pathway. So all of this basically activates cell growth, MAP kinase pathway, and this is about the receptor tyrosine kinase pathway. Now, insulin also works through receptor tyrosine kinase pathway. However, insulin receptor, this is the insulin receptor IRS, sorry, IR basically. This is also no, this is the insulin receptor, which has a alpha 2, alpha 2, beta 2. So these are the alpha subunits and these are the beta subunits of the insulin receptor. And insulin receptor in their case, after phosphorylation of uh, the insulin receptor, another protein known as insulin receptor substance or IRS binds as an adapter protein, and which again like leads to this PI3K or GRB2 pathway. Okay, so this is just an extension of the receptor tyrosine kinase pathway, which has an extra adapter protein. And for different insulin receptors, the IRS is actually changes. And we have several insulin receptors which have different roles and different tissue localization, and as well as the insulin receptor substances which have different localization. Okay. Next, we'll come to the JAK-STAT pathway. So, and for JAK-STAT pathway, so I'll also explain the JAK-STAT pathway. JAK-STAT. Okay. So JAK-STAT pathway is the pathway where most of the immunoglobulins actually immunoglobulin, sorry, not immunoglobulin, my bad. Uh, Immune signaling actually works through JAK-STAT pathway. Many of the immune signaling as well uh, as growth hormone or GH, prolactin hormones, they act through this JAK-STAT pathway, but it's mainly very famous for the immune signals. Okay, so how does this works? Again, is very similar to the receptor tyrosine kinase pathway. However, um, in this case, so this is my the receptors. So these are the GHR or growth hormone receptor, and this is my growth hormone. So this is my growth hormone. GHR growth hormone. So when GH comes, so there are two GHRs, and GHRs are bound with some is a protein known as Jack. So Jack protein. Jack protein is the full form of Jack protein is genus kinase, which name came from the Greek god Janus, which seems to be have gods of gates and doorways. Okay. Now, so the GHRs, so these are two GHRs. Now, what happens after the hormone comes and binds to the GHRs? So if this is my GHR, and the hormone has now bound to the GHRs. Okay. So now these jacks, they come close to each other. This is a jack and this is another jack. What they does is, so this jack, they come close to each other and they cross phosphorylate each other. Okay. So what is now happened? So this cross phosphorylate receptor as well as they phosphorylates the growth hormone receptor also. So now how the schematic looks like? Looks like this. We have a phosphorylation here, two phosphorylation here, as well as the Jack or Janus kinases. They are also now phosphorylated. Now, this is the growth hormone, okay? The GH. Now, this Jack proteins goes and binds to so these phosphorylated Jack proteins are now bound by a protein known as STATs. Okay, so this STAT, it's STAT. Okay, so this proteins are now bound by the STAT proteins. So these are the STAT proteins. Okay. So the STAT proteins, so the Jack proteins again phosphorylate and activate this STAT proteins. Phosphorylated STAT proteins now can form dimers like this. Okay. So the STAT proteins are forming dimers, and then this is my nucleus again. Inside the nucleus, I have DNA. These are now acts as the transcription factors, goes and binds and activates the gene expression. So the variety comes from the the combination of jacks and tags. So there are 4 jacks known as jack 1, jack 2, jack 3, and tick. And also there are around 8 stacks. So there are 7 total stat proteins. 7 stat proteins, which actually goes and which actually So the diversity comes from the different combinations of jack and. So this is the jack state pathway, and I have just described it here. So this is the jack state pathway. As you can see, the growth hormone is binding, and jack proteins are cross-linking to each other. They cross-linking to each other, and this cross-phosphorylating as well as self-phosphorylating. So then it's phosphorylates just stat proteins, and the phosphorylated stat proteins then goes and binds to some receptors and activate the gene transcription. So lastly, I'll go very briefly with the steroid hormone signaling. So as I have told you, in case of steroid hormone signaling, the hormone come inside the cells, go inside the maybe in the nuclear, because they also have in the nuclear receptors also. That's why the steroid hormone receptor is also known as the nuclear receptor. So they goes, it's a very simple scheme. They goes and binds to that nuclear receptor, which is in binds to the nuclear receptors, which then the inactive nuclear receptors get activated. And after activating these receptors form dimers with several cofactors, receptor form dimers and bind with several cofactors to activate the transcription, then proteins and cellular responses. Generally, the steroid signaling molecules, they will have this type of domain organization. So they will have an activation domain. Then they will have a DNA binding zinc finger motif, the activation domain 2, as well as the dimerization domain. So activation domain and activation domain 1 and activation domain 2, they are responsible for. So these two are responsible for hormone binding. The sorry, my bad. Sorry, I'm wrong. Actually, uh, sorry. This activation domain and domains are responsible for downstream signaling. This hormones bind to this here at the ligand binding domain here. So this is the hormone binding domain, the ligand binding domain, and dimerization domain helps in form the receptor dimer. Okay. This zinc finger motifs has the DNA binding domain, which helps in the bind to the DNA. So when there is a because these they acts as a transcription factor, so they should have a DBD or DNA binding domain. And it's also it's here that several proteins like HSP90, HSP70, and other uh, protein, they uh, inactivate the receptors. As soon as the hormone binds, these receptors get dislodged from the this proteins, this regulatory proteins get dislodged from the receptor, and the receptor gets activated and forms the dimer. So binding of this kind of regulatory protein prevents the unnecessary signaling as well as the unnecessary formation of the dimer. So yeah, so I have discussed many types of mainly different types of hormone signaling. I still could not discuss about notch signaling as well as the uh, nitric oxide signaling, INS inducible nitric oxide signaling. Okay, and also the soluble gluonyl cyclase signaling. Those there are many types of signaling that can happen. So also I cannot discuss the signaling of the uh, in the brain, which happens to the natriuretic peptides or ANPs and those. But yeah, but these are not, those are also very important, but these are the main things. And if you want to learn more about, then always go and please go and review this. Sorry, go and read this review. I request you to go and read this review where I'll give it there in the uh, in the NP the course folder also. So and then okay, and if it's and so now I'll ask if there was any question. So feel free to contact me at any point of time if you have any questions regarding this. So if there is no more questions, we'll stop the sessions here, and the next session will be the last tier session of this session course, and which will be kind of a summary session, which I'll take as a summary of all different types of whatever has been taught in this course. And yeah, so that's it. Thank you. Thank you for joining here, and good night.